Welcome to this second lecture in our series on understanding COVID-19. I'm Bruce Walker, and what I'd like to do now is teach you about how SARS-CoV-2 causes disease and how it is that the body fights back against this pandemic. To begin with, a short word about myself. I'm a physician. I was certain that I wanted to become a doctor taking care of patients, a family physician, if you will. This slide shows me when I first graduated from medical school and started my first job at Mass General Hospital. I was really excited to be there. I thought this is a place where they know every disease, and what a great place to do my internal medicine training. And it wasn't but a few months into that somebody came into the emergency room with a disease nobody had ever seen before. It was a person who ended up having multiple different cancers and infections at the same time. It was clear to me as a doctor that we on the front lines were going to be the first to see new diseases as they arose. And for that reason, I decided to pursue a different pathway than I originally envisioned, and that was to become a physician scientist. So physician scientists are people that really learn from patients by doing laboratory work and trying to understand what the causes of diseases are so that they can actually bring treatments back to patients. And I had these mentors here that really influenced me in making that decision. As time went on, we realized that in fact this new disease was something called AIDS. And it really took 5 years before we were able to define what this disease was and have a diagnostic test for it. In fact, it was 3 years, it took years to even know what the cause of this new illness was. So it was during that time that I began to try and study how it is that the body fights HIV infection. In the process of that, my career went in directions I never envisioned. We ended up doing work in Africa where there was a burgeoning expansion of the epidemic. We realized that there were things that we could do there by helping to build capacity and fight the epidemic locally with both science and medicine. We established a small laboratory, and then we were able to get funding to establish the first dedicated medical research institute to ever be built at an African university here in KwaZulu-Natal, as shown in this photograph. That led to us believing that we could have even more impact, and ultimately led to the creation of the Africa Health Research Institute. So now we and our African collaborators had a really substantial effort that could go forward, led by our African colleagues, but supported by our collaborations, and that allowed us to really make progress towards trying to come up with solutions for this pandemic. And in terms of trying to get people to understand that ongoing pandemic, I began to teach a course for Harvard and MIT undergraduate students called Evolution of an Epidemic. The course objectives were to use HIV as a model to understand how an epidemic emerges and get how clues from patients guide scientific discovery. And I show this slide because these are two of the individuals that were with me in January of 2020. One on the left was a student from MIT who had just returned from visiting her family in Wuhan, China. She started getting text messages from them about a new disease, a new pneumonia of unknown cause that was having devastating effects in the Wuhan area. On the right is a former trainee of mine, Diana Brainerd, who was at the time, the senior vice president for HIV and emerging diseases at Gilead Biosciences. She started getting phone calls from doctors in China asking if she would release a drug that she had developed for Ebola, another virus that's endemic in Africa, and that drug was remdesivir. So it was a combination of listening to those two people relate their experiences that made us realize that something really bad was happening with pandemic potential. And so as a result, as I was coming back in January 2020 to Boston, we decided that we would begin to work on this to try and apply what we'd learned from HIV to try and fight this new epidemic. was very quickly discovered that this new pandemic was caused by a coronavirus, so termed because of the outer spike protein that you see on this slide that has sort of a crown-like appearance to it. It's like other viruses. It's an outer coating that contains genetic code that allows the virus, when it infects a cell, to replicate. And the first question you have to ask is, why does SARS-CoV-2 cause pneumonia? And it turns out that for viruses to get into cells, which they have to do in order to reproduce themselves, they need a receptor, and different viruses use different receptors. And so in the case of SARS-CoV-2, that receptor is called ACE2, and it happens to be on lung cells and in cells in the nasal passages. And that's why the first cells that get infected are actually those cells in the nose and lung. What happens when a virus infects a cell is really pretty remarkable. Viruses have very few genes. In the case of SARS-CoV-2, it has about 10 genes. Compare that to the human cell, which has 20,000 genes. Those 10 genes of SARS are able to completely hijack the 20,000 genes in the human cell and reprogram that cell such that it no longer makes things that the cell needs, but instead it makes new viral proteins that then are encoded by the RNA that's injected into the cell. As those new proteins are made, what happens is new viruses start to bud at the cell surface and go on to be released and be able to infect other cells. So this is really the life cycle of a virus. It has this obligate period where it has to be within a cell, in this case a human cell, in order for it to be able to make new viruses. In fact, it's that very small amount of genetic material and that isn't able to encode for all the things it needs. And so what it does is it hijacks cellular processes to make them do its bidding. How does the body fight back from that? Well, it turns out that we have white cells in the bloodstream that are, some of these are called B cells. They're antibody-producing cells. And what these do is they are pre-programmed to be able to recognize different foreign pathogens that one may be exposed to. If that recognition event occurs, these B cells start making antibodies, and those antibodies then bind to the virus, ideally, and prevent the virus from being able to bind to the ACE2 receptor and thereby prevent infection. So this is the idea behind how the body is able, using its own host defenses, to be able to combat a new pathogen like SARS-CoV-2. And ideally, these antibodies will provide protective immunity or protection from subsequent infections. So on this slide, what you see is an uninfected person. If they get infected, what happens is the body starts to make these antibodies, and ideally, what happens is that the virus is completely cleared. You're left with the antibodies, and those antibodies then protect you should you ever encounter that virus again. Now, the problem with SARS-CoV-2 is that the virus infection itself actually causes immune system damage. So that impairs the ability of these white cells, these B cells, to make antibodies. And so you end up with a weakened antibody response. And this is something that we've been very concerned about how durable the protection will be from infection by the SARS-CoV-2 coronavirus. On the other hand, the body has other ways to fight back besides antibodies, and one of these is something called killer cells or cytotoxic T cells, and that's shown here on the right. What antibodies do, these B cells that are produced by B cells, is they neutralize the virus before it can infect a cell. You can think of these killer cells as kind of a backup. If a cell happens to get infected, the body has this incredible mechanism whereby it can alert the body that a virus is inside a cell and get the body to kill that cell very selectively. And the way that it does that is that these little viral proteins the viral peptide are shuttled to the cell surface as they're being made. From the virus's standpoint, it wants to make more viruses. What the body does is that cell takes a piece of that virus and shuttles it to the surface. And if that molecule on the surface, the host molecule HLA, has a viral peptide in it, the body knows that something bad is going on inside that cell. In come these killer cells, and they essentially kill the virus-infected cell at a time when the virus is particularly vulnerable because it's lacking its outer coating. And that leads to not only death of the cell, but death of the virus. So why does the immune system not do a better job? The reason for this is that, in fact, the immune system, the T cell response, actually becomes dysfunctional, and it starts to cause collateral damage. A killer cell comes in, recognizes an infected cell, and starts to produce these soluble factors called cytokines. What those do is they activate other immune cells, which activate other cytokines, and you end up with what has been termed an inflammatory storm, where basically there's a lot of collateral damage being done, and the killer cells themselves are dysfunctional and not effectively killing off the virus-infected cells. So here I'm showing you what happens actually in the lung and why it's so hard for people to breathe who become infected. On the left, you see a normal lung under the microscope, and you see these very thin air sacs, very thin linings of air sacs. In those linings are blood cells, and it's air exchange between the air sac and the blood cells that allows a person to actually get the benefit of breathing. On the right is a microscopic view of somebody who has SARS-CoV-2 pneumonia. And there you can see that there's this inflammatory infiltrate in between the green arrows that thickens that septum, those thin lining cells of the air sacs, and makes it much harder for oxygen to diffuse. And as a result of that, it becomes very hard to breathe. And so this is an effect not just of the virus itself, but an over-exuberant inflammatory response that's sort of calling in all the troops, but not doing an effective job in terms of getting rid of the virus itself. And so another way to think about this is that really the body's fighting against this virus can be put in two phases. One is an antiviral immune response phase, which is early on, where the immune system is really fighting to get rid of the virus. If it doesn't do that effectively, you enter into this stage where a stage really of collateral damage from the immune response, and that's this host inflammatory response phase where actually the immune response then becomes a foe rather than a friend. SARS-CoV-2 also causes other problems. The initial site of infection is the lung or the nose, or the cells lining the nose. But there are a number of other cells that can be affected by this coronavirus. Cells in the lung, there are certain type of cells called type 2 pneumocytes. There are cells in the gut, in the circulation, lining the blood vessels, heart muscle, and in the central nervous system in terms of nerve cells that can be affected by this. So the question is, we end up with these different manifestations of disease. One is that the infection of the nose and the infection there can lead to anosmia or loss of smell. The infection of the lung leads to pneumonia, of the gut leads to diarrhea. In the circulation, one of the manifestations of this disease is blood clots because of infection of those lining cells. And the infection of the heart cells can lead to heart attack. And there's infection of the brain in the central nervous system that can cause brain fog. So what is it that determines which cells in the body get infected by SARS-CoV-2? Well, think back to what I just told you about how the virus enters those first cells. It needs a receptor, that ACE2 receptor, and it turns out that all of these cells actually express that same receptor. The virus can't get to that until it gets into the bloodstream and gets distributed around, except for the lung and nose, where it can get in by breathing. And so that's really how we begin to understand why we're seeing the things that we're seeing in the emergency room and in patients that are hospitalized. And it really all makes sense because it's where the virus is actually doing its damage. So one of the other things that is really important to understand about SARS-CoV-2 is that it mutates, and I'm sure you're all aware of this. The question that I want you to be able to answer is, why do these mutations arise? Why would the virus be changing all the time? And if we think about this, it's really pretty simple. The genetic material of the virus is RNA. And when RNA gets reproduced as the virus goes through its life cycle, the virus has a tendency to make random mistakes. Those lead to mutations in the RNA. That in turn leads to mutations in the proteins of the virus that are encoded by that particular gene. And so what we're particularly concerned about are mutations that arise in the spike protein. So how do those mutations happen? What you can see here is what we talked about before. The virus enters a cell, new viral proteins are made, viruses butted off at the cell surface. But here's what happens with mutations, that initial RNA gets into the cell, it gets copied and a mistake gets made. Now that mistake is carried on to the protein product made by the RNA, so that the protein, in this case the spike protein, has a mutation. Now as new viruses bud off, they carry not just the mutated RNA, but also the mutation in the viral protein on the cell surface. And so what are the consequences of that? And here it's important to understand that there can be quite different consequences. On the left here, what you see is a mutation that actually leads to stronger binding to the viral receptor ACE2, diagrammatically depicted. In the middle, you can have a mutation that leads to a less infectious virus because it prevents binding as tightly to the ACE2 receptor. On the other hand, you can have mutations that are really irrelevant, that don't have any effect on the ability of the spike protein to bind to ACE2. So you can go from no change to mutations that make the virus less infectious to those that make the virus more infectious. And now you have to think about what the consequences of that may be in terms of which viruses are going to survive. If you end up with a mutation that makes the virus more infectious, it's going to win out and become the more dominant strain within a person's body. These mutations also can affect antibody recognition. Remember, it's antibodies that prevent the spike protein from binding to ACE2, and in that way prevent infections from occurring. And what we see here is on the left, we have an antibody that binds to the spike protein. Spike can't interact with ACE2 and no infection occurs. On the other hand, on the right, we have a mutation that abrogates binding by the antibody but doesn't prevent the spike from binding to ACE2. And so those are mutations that prevent antibody binding, so they eliminate the ability of the body to actually inhibit the virus, and yet the virus can still do its damage by infecting cells. And so those are those are really the consequences of these mutations. And the big question is, will antibodies produced by infection block these variants as they arise in the body? Somebody gets infected, each infectious cycle, every time a cell gets infected, in everybody that's infected across the world, it has a chance to come up with one of these mutations that makes it more infectious and that escapes antibody detection. Our hope is that the initial infection would generate antibodies that would protect against these variants as they arise, but that is something that we can't necessarily count on. And in fact, getting back to the issue that I told you about in terms of a more fit virus, a virus that's more infectious, more transmissible, becoming dominant. We have a great example of that in a virus strain called B117. So this is a SARS coronavirus. It's linked to the original virus that was first sequenced in China, but it has important mutations that make it more infectious. And what this slide shows is just that this has rapidly become the dominant strain. It overtook all other strains in England, and it has been spread now to countries throughout the world, and where it also becomes dominant. There are other viruses that are also emerging now that are becoming dominant, and I think you can now answer this question: why is it taking over? It's taking over because it has an advantage of being more infectious and more easily transmissible. That also changes the dynamics of the infection in any population where the virus happens to land, because now the same prevention measures like distancing and wearing masks, while they still work, they may work somewhat less well. And in situations where there's casual contact, the likelihood of getting infected then grows. So these new mutants are called variants of concern, and the first one was something called D614G. It was the first evidence that we had that the virus that we first detected in China, by the time it got to Europe and started spreading in Europe, it became completely different from this original Wuhan strain because of this one mutation, but all viruses ended up having that. That virus then emerged further as this B117 variant in England with enhanced transmissibility. And then in South Africa, again, more mutations and a new virus, this B1351, which also seems to be more transmissible. And the big questions here are, do these enhanced transmission, do they lead to higher viral loads and worse disease? And do they have an impact on vaccine efficacy? And we'll come back to some of those issues, but I want to just show you data from South Africa to just show you how this happened over time. If you look to the very left of this screen here, what you see is that there were two dominant variants in South Africa in January of 2020. This 20A version and the 19A version. But look at what happened to them as we go to the right over time. Those things essentially disappear, and they're completely taken over by a new South African mutant, this B1.351 variant. So the virus population is completely turned over. Now, if somebody got infected in January of 2020 with the 19A strain, would they be protected against the mutant strain? That's the concern. And the good news is that there is some cross protection. The concerning news is that it's less robust, and we'll get back to that more in the final lecture of this series. One question I'd like you to think about, though, is why is it that there seem to be many of these new variants arising in Africa and Brazil? Well, one of the reasons that we think that may be the case is because, now getting back to thinking about the HIV pandemic, there are lots of cases of HIV infection inadequately treated in South Africa and in Brazil and in other countries. If you have a situation where people are immune suppressed already, either because of these other infections they have, like HIV, or maybe because they've been on chemotherapy, what can happen is it can be much more difficult for a naturally acquired infection to be cleared by the body's immune system. So those antibodies are less robust, the T cell responses are less robust, and you end up then with more time for the virus to replicate in the body, more cycles of replication where it can make mistakes and go on to become more transmissible or more lethal. So I'll wrap up now from this second lecture by just summarizing a couple of the key points. One is that viruses need cells in order to replicate. They carry very little genetic material, but they use that genetic material to basically commandeer host genes and host pathways to force them to make viral proteins for them. Those go on to bud at the cell surface and go on to infect other cells. The body fights back against this by developing antibodies, which are produced by white cells in your body. Those antibodies are able to neutralize free virus. There's another arm of the immune response called killer cells, or these cytotoxic T cells, that are able to kill virus-infected cells by recognizing viral proteins on the surface of an infected cell. The immune system is both friend and foe in this infection. Early on, it tries to fight the infection. If it doesn't get the upper hand in a timely way, this can sort of balloon into a lot of collateral damage and actually a worse outcome. And finally, we're seeing emerging variants that clearly seem to be more transmissible. And I think this underscores the fact that we need to do everything we can on a global level to stop this pandemic and get as many people immunized as possible so that the virus has less chance to mutate, less chance to get the upper hand, and ultimately lead to an end to the pandemic. So thank you very much.